We are here at the Auditorium di Santa Polonia in Florence in the framework of the final General Assembly of the BIOSYSMO project and right before the start of the Bioremi 2026 conference. We are here with our coordinator, Dr. Sara Gil Guerrero from IDENER. Hello, Sara.
Hello, Joanna.
Could you briefly describe the BIOSYSMO concept and the main outcomes?
BIOSYSMO was conceived as a multidisciplinary framework that brings together environmental characterization, microbial ecology, omics modeling, all to support the development of different biotechnological technologies that aim to tackle complex mixtures of contaminants in soil, water, and sediment. The main idea behind this is that the realistic scenarios in which pollutants are present are not as a standalone, and from this mixture comes the complexity of really deploying effective systems. In the course of BIOSYSMO, we have had significant advances. In particular, we have generated a large amount of both environmental, biological, operational data that has served to identify which are the key players in the field. And with that, and also supported by a model-driven framework, we have been able to support the design and development of different technologies. In particular, in the scope of this project, we have developed 6 cases of study that tackle different compartments and pollutants, and also that cover microbial consortia, plant microbe systems, and also bioelectrochemical systems. Altogether, BIOSYSMO has been able to demonstrate how a mechanistic and model-driven design can support the effective development of these technologies that later can be transferable to other type of sites.
Great. I would like to welcome Dr. Lila Otero from IDENER, member of the coordination team. Hello, Lila. How does digital modeling and system integration within BIOSYSMO support more efficient and scalable bioremediation solutions?
So in BIOSYSMO, we develop a computational framework that supports the bioremediation innovation at different stages of the process. So first, we created a database that integrates different data of microorganisms, enzymes, and other resources useful for bioremediation. And with this database, we supported the search for traits that are beneficial for bioremediation in different microorganisms and communities that were being developed by experimental partners. In this way, we help them to rank these microorganisms and prioritize them to further studies, supporting them in these early stages where you have different resources and you have to select which ones will go further. Then, once they made this selection, we incorporated metabolic models that support the modeling of how the metabolism of these microorganisms work. And very interesting, we could simulate how the different players in the communities interact with each other, what metabolites they exchange, and what interactions are important for these microorganisms and microbial communities to work. Finally, another stage where we supported the bioremediation process was during the technology development. We incorporated process models at the technology level, so we can see how different operational parameters affect the efficiency of the bioremediation. We can simulate other scenarios, so that also helps to extrapolate the results to other scenarios, and we also support process assessments such as estimation of cost or life cycle analysis assessments for environmental impacts. And so overall, we develop a framework that covers different stages, and also we apply it for different technologies such as phytoremediation, bioelectrochemical systems, and bioaugmentation. So in this way, we develop a framework that we validate with different technologies and is robust to support the experimental development to avoid trial and error, and applicable to different projects.
Great, thanks. So we are here with Dr. Alfredo Perez de Mora from TAU. Hello, Alfredo.
Hi.
And I would like to ask a question to you. So from an implementation perspective, how do real site conditions influence the validation and scalability of the BIOSYSMO technologies?
Well, I think that's a very important aspect to consider. And in BIOSYSMO, we had an approach to deal with that. So the site influences very strongly the potential technologies that will be used there. But we did two things. First one was to work from the very beginning with site materials so that the microbes and the potential consortia or enrichment that were obtained, so the biosystems were already used to the future site conditions. And on the other hand, we also developed an approach considering different key performance indicators so that we would be able to evaluate the different technologies using a different frame of indicators to see whether or not they're ready for field application. And then, of course, even if the technologies work at a very fundamental level in the lab, there are things that you have to take into account at the site. And these are site-specific, which could be starting from health and safety aspects. It could be also related to the heterogeneity of the site and also to the availability of the location. So these are the site-specific things, but also you have to consider that if the technologies work at the lab, whether they would make it to the field also depends on economic resources, and if the partners are able to scale up those technologies, because the setup that they will have in the field will be different from the lab. So it certainly does, and this is the reason why in the end, from all the different technologies that were developed at BIOSYSMO, only 2 of them were chosen for pilot field scale demonstration.
Thank you. Hello, Edward. We're here with Edward Porras from LEITAT, and I would like to ask you a question. So what advantages do bioelectrochemical systems bring to the treatment and recovery of contaminated water within BIOSYSMO?
So bioelectrochemical systems as like general group of technologies bring a clear advantage, that is the reduction of reagents consumption and the reduction of energy consumption per amount of pollutant treated or removed from, let's say, a matrix. It could be a groundwater, soil, or whatever. And in the case of BIOSYSMO, we have been working on the removal of 3 types of pollutants with the use of bioelectrochemical systems. Initially, we worked with metals and metalloids removal, so we could make some great advances on this regard compared to the previous knowledge that we had from at the beginning of BIOSYSMO. We even mixed or coupled the bioelectrochemical removal of metals with phytoremediation processes together with the University of Burgos. The second main technology was the removal of hydrocarbons from groundwater coming from an industrial site. We could make some advantages in reducing the need for oxygen in the process, so that we can, at the end, I think that after the project completion, we can foresee that we can implement the technology right in situ, right in the proper aquifer. But it will come later on after BIOSYSMO, but we made some great advantages in that direction. And finally, we have been working on chlorinated ethanes removal. Our main idea here was to tackle 2 questions on this topic. how we could supply reductive power to promote the dehalogenation of these compounds, and how we could work in a reactor that is membraneless, meaning that there is no need for a membrane inside the reactor. And we could tackle all these challenges within the frame of BIOSYSMO.
Great, thank you. I would now like to welcome Professor Michel Chalot from the University of Marie and Louis Pasteur, one of the BIOSYSMO partners. Hello. So I would like to ask you one question. Can you describe UMLP's role in BIOSYSMO and how plant-based systems contribute to sustainable soil remediation?
Yes, surely. UMLP was in charge of one major work package 3, on the biosystems, how we produce the biosystems with plants, with microbes, including fungi and bacteria. And also UMLP was responsible for providing a case study site. So we had 3 sites overall in the project, located in France, and we collected some soils, we collected microbes from there, and we were able to interact with the other partners from the project to give them the opportunity to work with these microbes, with these soils, and to better understand what is going on in this particular situation, because all the soils that we collected from the French site are contaminated with metals and PAH. We hope with finding the right microbes to be able to improve the growth of the plant that we aim to develop on this site, because the sites are really, the soil is very difficult for the plants. So we hope to improve the growth of the plants using these microbes that have the capacity to cope with the heavy metals or to degrade with the pH.
So we're here with Professor Blanca Velasco from the University of Burgos. Hello, Blanca.
Hello.
And I would like to ask you, how is University of Burgos advancing plant-based water treatment and bioaugmentation and biosimulation technologies within BIOSYSMO?
Okay, thank you, Joanna. Yeah, I will briefly explain. A part of these core technologies that I will explain now, we have been transversal partner, for instance, with the toxicological analysis performed before and after the implementation of the remediation strategies. But let's say that our core technologies have been plant-based, as you already mentioned, and in this sense, we have been very successful in integrating this system together with Leitat colleagues, with a hybrid system in which, or thanks to it, we have reached and overpassed the KPIs, reducing the metalloids contamination in a very nice manner. I have to mention that this system comes from previous projects and it was awarded by the Innovation Radar of the European Union. In this system, we took advantage of the PGPR characterization performed within BP3 also in the project by our colleagues from UBU. And regarding the other core technology, it's applied to soil contaminated with some organics like hydrocarbons, lignine and atrazine as well. We applied several strategies on biostimulation and bioaugmentation. We have been collaborating quite closely with GSI and also currently with CNRS, and we have also achieved quite nice results regarding biodegradation of these contaminants and overpassing in some cases the KPIs. So we are quite grateful of these results, although we keep on getting new data and we will go beyond again.
Great, thanks.
We are here with Ana Paula Mucha. She's a researcher at CIMAR and she leads the Bioremediation Microbes for Sustainability team. CIMAR is a partner of the BIOSYSMO project, so Ana Paula, what was the role of CIMAR in the project, and also how do plant-microbe interactions help restore contaminated storage environments?
The role of CIMAR in BIOSYSMO was the development of microplant biosystems to recover and also to remediate estuarine environments. We start by isolating microorganisms from estuarine environments in order to obtain specific strains that can collaborate among themselves to remove contaminants and also have a positive interaction with plants. After this initial characterization, the more relevant microorganisms that present complementarity were organized in microbial consortia that were, after these microbial consortia were tested in microcosm systems in laboratory environment. After selecting the most effective microbial consortia and their interaction with the plants were characterized, we moved to a larger scale experiment that was a mesocosm experiment in greenhouses, where we also simulate the estuarine environment by simulating salinity and also, for example, tidal cycles that occur in real environment. This is very relevant because estuaries are very challenged for organisms, so organisms have to be adapted to changes in salinity, changes in water level, oxygen. So these estuaries are very dynamic systems. Also, most of the estuaries are very impacted by anthropogenic activities. So most of or large part of salt marshes may have been lost. So also it's important that these remediation strategies combine bioremediation with revegetation. For this, the interaction between microorganisms and plants are very relevant because they allow to increase the capacity of the plants to survive, and also plants help microorganisms in their bioremediation activity. So this synergy is particularly relevant in the estuarine environments, and that's the overall aim of this of our contribution here in BIOSYSMO, is to develop this biosystem that can increase water and sediment quality and at the same time contribute to ecological restoration of the estuarine environment.